for that. Okay, so I'm Ben Curran. I'm a PhD student in Ghassan's lab, also working with Dr. Nabi at UBC. So my very short presentation here will be introducing our work on super resolution microscopy. We deal with two use cases in a single application. In the left-hand side, you see a cartoon image of a prostate cancer cell, and at the bottom you see the membrane, which allows the flexibility of the membrane, allows the cancer cell to have motility, which then allows metastasis. So this mechanism is controlled by small spherical invaginations called gavuli, about 100 to 150 nanometer large. So at that size, you need super resolution microscopy to actually see the dynamics of that mechanism. Below, then you see the actual image of such prostate cancer cell. And then the small inset shows you the structures at 50 to 100 nanometer wide. What we want to have is for those images, this is a 2D image, but we want for those images in 2D, but then also for temporal sequences to automatically identify that mechanism. So in green, you have the scaffolds, in red, you have the gavuli, and then in orange, you have the structures in between. If we can capture that, we can capture, hopefully, the dynamics of metastasis. The second use case is very similar. We have here confocal microscopy of retina tissue of Alzheimer diagnosed patients. Here we're labeling for amyloid beta. We know that those deposits are necessary but not sufficient for Alzheimer diagnosis. So we want to obtain exactly which deposits are indicative of Alzheimer and which are not. For example, in healthy tissue, you also have amyloid beta presence. So more formally, in computing science problem statement, we have an image I and an image level label L. We want to find all the objects, and for each object, we want to compute the statistical evidence the object has in support of the image label L. We want to do it in an unsupervised setting. We have no information. Indeed, the task is to discover the information. We also have the problem that the images can originate from different microscopes, taken at different times, different operators, and so forth. It should be automatic, so completely self-tuning, and the label should have its own encoding of uncertainty and have a non-trivial theoretical limit. So the label variance should be constrained to a certain extent. So then we have here a very dense figure from our paper that gives an overview of how we go about this. In the top, you see a schematic of how those gavuli form and deform under stress. The flowchart, I will deal mostly with the labeling part, but the flowchart here shows how we do the object detection and then use belief theory to enable the labeling. And then below you see how for those objects we obtain the individual labeling. We have a 3-color scale here where we assign each object with a possibility of backgrounds, scaffolds, and gavuli. So then what's belief theory and why do we use it? It's a superset of standard probability theory. So anything you can do with probability, you can do with belief theory, but the opposite does not hold. It's more flexible than probabilities. More formally, belief theory introduces a plausibility, namely the maximum support for a proposition, the minimum, namely the belief value, and then also the uncertainty, the difference between plausibility and belief. How do we link all of this together? Well, for a certain proposition, namely the encoding of a certain statistical evidence that an object has for the label, for example, the label can be Alzheimer or metastatic cancer. We can compute the normalized statistical distance in feature space to the object distribution. We can then use Cantelli's inequality to obtain a maximum probability value, which then gives us our plausibility label. And then using the Dempster-Shafer calculus, we can actually derive the belief and uncertainty values from the computed plausibility label. Belief theory is quite complex. I have the references in these slides if anyone is interested in looking into more depth into this. So then, does this work? Well, first of all, we know from the related state of the art that those cavuli actually have a frequency of about 20%. We see that that frequency cutoff, when superimposed on our cumulative distribution function, coincides with the elbow point, which is consistent but not sufficient validation. We also know that cavuli only form in the presence of another protein, namely P2F. And here we see that the presence of P2F markedly increases at the same, roughly the same point, which then validates the approach. I mentioned heterogeneous data sets and conflicts. So this is an example of how we use that for the Alzheimer use case. We have here on the top left a single image, again, of amyloid beta. The suspect points here are highlighted. We obtain this label using two different data sets that from two different microscopes, even with different pixel precision settings. But using belief theory, we can actually formulate quite intuitively how to combine those models without having to retrain or recompute the belief and the probability values. Second, if you follow this approach, we can also compute the conflict, namely the disagreements with between the data sets. This is shown in the last slide, where each object here is, each point here is an object in an image. And we show here for 3 images the weight of conflict. The higher this is, the more discrepancy there is between the two models coming from different data sets. And given this, you can give this back to the end user, and then they can report their findings with hard theoretical limits and uncertainty about their findings. And that's it for me. I would like to thank the advising professors, my very patient co-authors, and everyone from the labs at SFU and UBC for all their advice and support. Are there any questions?
Okay, everyone. So we have around roughly 5 or so minutes for questions. If anyone who wants to have a question could either type into the chat or raise your hand to be unmuted, that would be fantastic. Okay, we see we have a few.
Do I stop sharing or?
Oh, you can keep sharing. Hold on.
Okay.
Can I
yes,
ask?
Alejandro.
Yeah, thanks. Thank you for the presentation. Can you go back to the methods? One of the methods slide, please. I don't remember which one. I think the one before that one.
This one.
Yeah. So, I'm not sure if my question is a dumb question, but just like why is it constrained by like unsupervised methods? Why not like supervised?
Because the question is literally about discovering what each object in the image is. So if I go back here, I'll try the annotation. So there is no annotation for this because this is literally going to be reported in paper saying, oh, this object is X. In theory, it is possible to label some of these objects, like semi-supervised maybe. But we felt that given the task of like pure scientific discovery, it was better just to approach the problem from completely unsupervised. that being said, at the image level, so for each image, we do have a label for that image. So in that setting, it is supervised. Does that answer your question?
Yep, that makes sense. Thanks.
Okay, it looks like we have time for one more question at least. Let's see. Okay, so I guess if no one else has any questions, we can go ahead. Oh, wait, here we go. Looks like there's a question in the chat from Madeline. Okay. So Madeline asks, she was curious whether or not you've tested denser images and how it went, i.e. images where the cells are much closer together.
Yeah, that's a very good question. So in this in the Caviolon 1 setting, we only have one cell in the image. In principle, it doesn't really matter if there's multiple cells in view. Although there might be an effect where, for example, in fluorescence microscopy, you always have the case where, for example, one cell would have very poor labeling, and the other cell might have better labeling, or for example, one cell is actually not viable, and so forth. So that can affect the end computation of the probabilistic label. But that said, usually we can segment out the cells to, for example, test if that's the case. For the and I should have made that more clear. In this case, we're looking at tissue. So this is like you're basically, those deposits are between the cells. So here you're looking at hundreds, if not thousands of cells. I'm not sure if that answers your question.
She says yes. Okay. So I think barring any very, very short questions, I think we will move on to our featured talk for the session. If Ben, you could relinquish your screen at this point. Sure. And then if Zamin, if you would like to get your slides up.
Sure. Hi, everybody.
Yeah. So I'll just give you a short introduction. So, yep. Zamin Iqbal is a group leader at EMBL, as I stated previously, the European Molecular Biology Laboratory at Cambridge. He leads a team that works on fundamental method development for microbial sequence analysis. And today he's going to be talking about cryptic variation in the bacterial accessory genome. Everybody, please give a silent hand for Zamin. Thank you.
Hi, everyone. I've just realized that I put the wrong title on my slides, but don't worry, it's the right talk. Thanks for coming. As I've just been introduced, I'm a method developer. I used to try and work on ways to sort of discover and genotype genetic variation without using reference genome to try and get rid of bias. And over the last 5 years, I and my group have been working on methods. Instead of using no reference genome, I want to use sort of all the prior knowledge we have about a species and try and do better, basically, at studying genetic variation. So this talk basically starts out with some biology, trying to explain what the problem is. And then there's a bunch of technical stuff in the middle, which you should just sort of just let your mind float and just watch and listen and try to follow along. If there are bits that you don't catch on to, don't worry, because I come back to them. And then the last chunk is all looking at results and trying to see what this means. So I'm going to talk about how bacterial genetic variation is sort of fundamentally incompatible with using a linear sort of single reference genome. This is not news really to microbiologists. I'm going to outline an approach, give some details, and then evaluate it essentially. I can't start this talk without emphasizing how much work's been done by people in my group. In particular, this woman here, Rachel Cahoon, was my PhD student. She introduced essentially like all of the major concepts in this talk. She's on maternity leave, so she couldn't give the talk instead of me. And also Leandro did a huge amount of work on this, Kerry and Michael in particular as well. I'll reintroduce them as we go through the talk. So, okay, so first, I'm sorry if you know all this, many of you will know all this, but I think it's worth just emphasizing the things that are on my mind. Oops, let's try that again. Here we go. Okay, so bacterial genomes are gene rich, okay? Genes cover whatever, 85, 90% of the genomes. And you can therefore, sort of as a side effect, you can use shared gene content as a sort of proxy for genome-wide similarity. And so 2 E. coli, I mean, it really depends where you pick them from, but they may share maybe half or 70% of their genome and therefore their genes. Now, it really depends who I'm talking to, but for some people, it's a shock, if you see what I mean, that's so different from human genomes where, you know, even though the genomes are bigger and they have big structural variations, like if I pick you, any of you and me, I mean, the amount of alignable sequence between us is huge. So bacteria are very different. But the trouble is, of course, many bioinformatic tools are developed, you know, with the human genome in mind. And a universal pattern that seems to be seen across bacteria is this frequency distribution of genes. So if you take a set of genomes from one species, then you have this characteristic sort of U-shaped curve, and it may be more peaked at the left or at the right. This is sort of a fundamental thing we have to sort of cope with when we think about whether we use a reference. And so other factors, I'm just going through some factors we need to bear in mind. So genes sort of kind of appear and disappear in blocks. This is just each horizontal row is a different genome. It's the same spot in all of these genomes. It's an insertion hotspot, and each colored block is a module of, you know, multiple genes. And so you can see, you know, the same, you know, this block appears here and here and here and here. So you can get the same collections of genes sort of inherited in blocks. So how should we do SNP calling in bacteria? That's the problem I want to sort of address. How can we cope with these chunks of genome that shared only by subsets? And everything I'm going to do and everything I'm going to evaluate is working from sequencing reads. But the problem remains even with perfect assemblies. Like even when we're getting like amazing single contig assemblies, the problem of how we sort of describe and represent these SNPs is still there. So here's a toy example. I've got 6, as I'm sure you can tell, these are 6 E. coli genomes. Each of these colored regions is a gene. It doesn't really matter, it could be an intergenic region, but so let's say for the moment they're genes. So these green things here are all the same gene in different positions in these different genomes, and the numbers are just SNPs. And then you ask yourself the question, well, if I choose one genome as the reference, which SNPs can I call? And it's pretty obvious you can only call the things that are on these backgrounds. So anything on, you know, this gray gene is not going to be callable. And in this toy example, you can sort of, it's a toy, so the numbers are meaningless, but you can count up, you know, what proportion of the SNPs can you call using that as the reference. And that kind of evaluation is what I'm going to do later on with some real data using our method. So I'm going to introduce 2 concepts. I think they're quite handy. One is sort of hard reference bias. So this means if there's a huge chunk of genome that's missing from a reference genome, in other words, everything I've talked about so far, then you're going to have hard reference bias. Whichever genome you choose, there's stuff missing from it that you can't address. And that's an inescapable consequence of this plot. Okay, there's a lot of rare stuff. Everybody's got rare stuff in them. It's going to happen. There's a second thing, which is sort of soft reference bias, and that's basically refers to you as the edit distance, as the genetic distance between a sample and a reference increases, it becomes more and more difficult to map reads, and it becomes more and more difficult to align and to call variants. And this is usually the argument that's used for needing a graph genome. You want to remove what they call reference bias. But usually people are really talking about soft reference bias there. And for bacteria, you need to worry about hard too. Okay, and so these are 3 people who did most of this work. Rachel, this was her whole PhD. Michael, this is 2/3 of his PhD and still going. Leandro has been working like hell for the last year. So this is our approach. Some spoilers about what we're going to do, just to make you stay on the call. I'm going to describe the representation. So this is a type of graph called a minimizer DAG. Then I'm going to talk about how you do a single sample inference, so how to get one genome out of it. And it's actually a 2-stage approximation. And then I'm going to talk about how to do multi-sample comparison. And all of them require, there's the whole sequence of dominoes I need to get to fall on top of each other. So I'm going to describe a whole sequence of different things. Don't panic if you lose track of some of it, because I'm going to come back and go over it again. Okay, so the first step, this is our strategy. Genomes evolve by recombination and mutation, no shock. So new samples should look like sort of noisy recombinants of old ones, if I've got a bank of old ones that I remember. So what we do is we start with a reference panel of genomes in such a way, we set them up in some way that we can map to that reference panel and allow reads to map across recombinations. And then somehow that mapping is going to tell me what, have I got a better, I'll show you this in 2 steps. The first step is, given reads from my unknown genome and my reference panel, which I'll explain what that means, I'll find the closest recombinant or mosaic of that reference panel to my unknown genome. So the first step of my approximation is to find a recombinant that's like it. And then the second step is I fix up any errors that are left with local assembly. And I completely avoid, for those who care about graph genomes, I completely avoid actual genuine alignment with cigar strings and all the rest of it. I don't do alignment to a graph because that's hard work and really for our purposes unnecessary. Okay, so that's the spoilers. I'll go on to step 1, this representation. So we define a thing called a pan-genome reference graph. It's an unordered set of directed acyclic graphs. Okay, I'll explain that. So each graph represents some orthology block. It might be a gene or an intergenic region, it might be a mobile element. But for the examples I'm going to talk about today, I'm working on E. coli, and I've taken the genes and the intergenic regions and I've separated them out and built graphs from them from multiple sequence alignments of versions that we've seen before. We completely let go of higher order structure, the ordering of the genes and things like that. That information is not stored in the graph. So what does this mean? Here's a multiple sequence alignment of a set of, or at least it's an alignment of multiple genomes. And it's not very obvious actually looking at this, but they are slightly different. But I can lay them out like this. So they all start with ATC. Then you either have GT or TAT. And if you have GT, you can either have a C or an A afterwards. And then they all end with GGA. So it's just a graphical way of laying out a multiple sequence alignment. And in order to enable mapping and search, we normally want to index it so that you have a read and you can sort of figure out where it goes. I'm going to skip this section because actually it's not worth distracting us with. And having got my multiple sequence alignment, I need to find a way to build a graph from it that sort of captures when genomes are similar and when they're different. What I want to allow is sort of what I call nesting. So I want variants on top of variants. For example, if there's a long haplotype which is quite different, I want to be able to notice that there's a SNP on top of it. Or I might want to either have a deletion or one of several SNPs. And I want to avoid this thing called blow up. So people who build genome graphs often have this problem where sort of memory sort of explodes because there's too many different, you have too much clustered SNPs and the number of potential haplotypes goes up exponentially. So we want to avoid that. So here's what we do. Just visually, I'm going to give you the algorithm. We start with a multiple sequence alignment. So I've got, this block represents my alignment. And then I look for vertical slices in my alignment of length, some length, say 7 base pairs, where they're all identical. So in these 2 yellow slices, all of those genes are identical. And in blue, I'm not saying anything yet. I'm just saying that in yellow, they're all identical. This slice is all the same and that slice is all the same. So what I'm going to do is I'm going to collapse those yellow things because they're all the same. So I'll collapse them down to a single representation. And now the 3 separated blue chunks I can deal with completely independently. And in each of them, I'm going to use k-means clustering to reorder the rows. So what I want to do is just cluster the rows so that they're more similar, like this. So these guys are similar, these guys are similar, and these guys are similar, and so on. And now, having done that, I can look at each of those blocks independently. This is a recursive algorithm, and do the same thing. I look for vertical slices inside each of those blocks, and then I do k-means clustering again, and I keep going. So let me show you a video of that working on some sequence. It's just a toy. But first I find blue stuff's the same and I collapse it. Then I find red stuff's and oh God, that's faster than I can talk. I hope you could see that. It's just, all you're doing is looking for similarity and collapsing, and then within the regions sort of clustering your similar things and collapsing again. And then, so there's a chunk in the middle which is quite bioinformatics heavy, and we're into that now. So there's a notion of it, what's called a WK minimize over string. What does that mean? So for any sequence, I take all the k-mers, so words of length k, starting in a window of length w, and I choose the least one. And why do I do that? This is just a way of systematically selecting seeds. And I replace my original graph with a graph of the minimizers. So in other words, I have the same graph shape, but I just use those seeds scattered across the genome. So what I've done is I've taken my multiple sequence alignment, I've turned it into a graph. You can actually, okay, if this was a real talk, if this was a talk with you guys all standing in front of me, I'd stop now and get you to bug me, but I think that's not going to work here. So I'm just going to say for the moment, I hope you can see that that's a way of representing a multiple sequence alignment. Because I've scattered seeds or k-mers across it, I'm going to be able to do some kind of matching with my reads. And I haven't said how, but I'm going to use this to find recombinants. So let's do that. This is my whole workflow. I build my multiple sequence alignments for each of my genes and intergenic regions, and I put them in a big pile somewhere, and I call that a Pan-genome reference graph. I index them, and then what I'm going to do is each of these squares represents a different sample. I want to analyze a bunch of different genomes, and I do them independently. And for each one, I sort of map. It's not quite alignment, but I do quasi-mapping of my reads to the graph. I work out where the reads align to. I get a mosaic approximation, a genotype. I do de novo assembly. I'm just giving you an overview here. And then having done de novo assembly, I find new stuff, and I go back and put it back into the graph. And I go back through the whole process again. And at the end, I'm going to be able to compare all of my samples. So in the middle, I'm parallelizing by sample because they're all independent. And then having done that, I can parallelize by gene, but have multi-sample genes. Okay, so here's a perspective. So reference-based variant calling is sort of a first order approximation. You basically assume your sample is the same as the reference genome. You map your reads, sort of pile them up. You isolate positions where the reads obviously disagree in a consistent way with the reference, and you say that's a SNP. You fix those and you're done. Roughly speaking, that's how it works. And it's a first order approximation. It works when it works. It's terrible for indels or for clustered variation because things become messy and ambiguous. Pan-RG-based variant calling you could think of as a second order approximation. The first thing you do is find a recombinant of your previous genomes that best approximates your sample. And then you say, that's a good reference genome. Now I can map my reads to that. I can find new things. We actually do it by local assembly. And then you go from there. And those ideas have been around for a while. They were used in plant genomics, in the MHC in humans, in Plasmodium falciparum, and again in humans. I mean, the idea's been around for a while. So what do? We sketch a read. I'm going to show you this pictorially in a second. We sketch a read and collect these minimizers. That means these representative k-mers scattered across the read. My index tells me which genes have those k-mers, where I've got matches. And for each k-mer shared between a read and some gene, I define a hit as the coordinates of where that is in the gene. And then I do some filtering and I look for clusters. I'll show you that. So here's my genes represented as graphs. I can turn them into k-mers. Here's my read, and I sketch it in the same way. And then I look for matches red guys are sequencing errors. And over here, you can see this. I've got a collection of matches with that gene, a collection of matches with this gene, and a collection of matches with that gene, and red. And I've got a lot of red mess. So I should have, I mean, the method is sort of neutral, but in my mind, I've been really very much thinking of making this work on nanopore data. So it needed to be able to cope with a lot of errors. So we look for clusters of k-mers shared with a local graph with one of the genes clustered together on the read, and that's how we work out that the gene is present, and also how we collect data on the gene as to like where you've got coverage. And then we do this thing called mosaic inference, where here's our toy gene, and you've just got sort of one difference here, and you notice all your coverage is there along the top. So it's sort of obvious there, but obviously the real gene graph's a bit more complicated, so we use dynamic programming. And all we do is use that to assign a maximum likelihood path, which is our first order approximation. It's not the consensus of the reads, it's the closest path in the graph. In other words, it's the closest recombinant of the genomes we put in at the start to the genome under study. That's our first step. So having done that, we found the closest path to the sample. It's inevitably not going to be quite right, okay? I took a bunch of genomes as my reference panel, but they don't have all SNPs in them, they just have a bunch. So there are going to be some SNPs and indels in the sample that are not in my graph, and they're going to be signaled by gaps in coverage. So if I look along my graph, and my blue line is my coverage along the gene, and you can see the coverage collapses here and here and here. And the green lines are the green sort of slices or ribbons are the places where we've detected coverage drops. The red dotted lines are actually places where there are genuine SNPs. And so it sort of works. It doesn't always work. At the end of the gene, of course, you can actually have a drop off in coverage too. But what we do is we locate those regions and we do local assembly. And unlike normal assembly, where the goal is just to get the genome out, we're quite happy just getting a bunch of possible options. We just sort of walk the graph, find a lot of candidate options, and we put them back in. And a later step is going to choose the right one. And this is the work of my student, Michael. So what we've got is we've got our reads. We've done a local assembly. We found that this orange path was missing from the graph, so we put it in. And when you remap the reads, now they've got somewhere to map to.
Hi, Sam.
Hi there.
Yeah, we can, if you would like to, we could just poll the audience for stopping for questions if you would like to see, if you can clarify.
It's a good idea. Thank you. Yes, just in case anyone, it's a good idea to, if anyone has anything to ask, now's a good time.
Yeah, so just like you have normal questions, if people could just raise their hand and we will unmute you, or we will look to the chat. I have seen a lot of questions in the chat about weighting of the graph you start with, if it is weighted.
It's completely unweighted. So in other words, I think you're asking me that if the same sequence was seen 20 times in my original reference panel, would it be upweighted by 20 or something like that? And the answer is no. So all we do is, if something's seen twice, it's just represented once. It's supposed to give you the breadth of variation you have in the species, but it doesn't have sort of frequency information in it. That's an interesting question. I might come back to it. We okay?
Okay, Alice?
Hi, I was just wondering if you'd considered whether there's benefits to indexing the read set rather than the graphs themselves, in case, you know, you want to re-perform the analysis later with different pan-genome graphs.
That's an interesting idea. So you could definitely do it. You could definitely do it. I guess it's a question of where you're coming from. At the moment, I'm thinking in terms of, will become a bit more obvious as we go through, but the problem I'm trying to get at is I've got a cohort of samples. I've got, I don't know, 100 E. coli or something like that, and I want to compare them. And what I want to get out of it is, I want to get sort of like a matrix where the the rows are different genetic variants in genes, and the columns are all the samples. You sort of want a full matrix, making a statement about every sample at every SNP, say. And so
you
that
could also then add which SNPs you're searching for and refine your search if you've indexed the read sets individually, rather than indexing the graph. And then when you add, you know, your orange path to the graph, you're not.
you still need to index the graph.
Why?
Because
you're
you need to be able
just
to map.
extracting k-mers from it.
No, we're doing a bit more than that. Apart from, so, okay. This is not a k-mer detection and presence absence thing. Because of the dynamic programming, it really does matter like where they are in their ordering. And if you're going to, in addition, if you're going to map to, if you're going to map nanopore reads, where basically really a ton of the k-mers in your reads have got errors, okay? One of the ways in which you're able to do that is because you can map the read to across multiple genes, you can figure out you can basically figure out where the read comes from, and therefore not exactly error correct, but error remove. So
I guess I was suggesting creating an index that's a full text index rather than a k-mer based index of the reads.
So the point I'm getting at is a full text or a k-mer index of the reads is a valuable thing, okay? And for the ends to which for the goals which I'm trying to achieve, I really need the graph. I need the graph of the genomes as well, precisely because I'm trying to find these recombinants and I'm trying to compare these genomes. I can imagine using the full text indexes of the reads as a way to accelerate that. But why don't you wait until you've seen the rest of the talk and you'll see why I'm trying to do it.
Sure, sounds good. Thanks.
No problem. Anything else?
For us?
Thanks. I mean, the original question I asked about weighting of the graph is related to the complexity of the DAG you're building, right? For a low allele frequency mutation, you're just adding another node and two edges, and I'm guessing that this is in further down the line, you are adding like the dynamic programming on top of this, right? So simpler graph. That would be, even if it shows in your sequence, that it'll be a simple mutation in one of other paths, right? And simply can show it as a null one, although you have seen it in other genomes before. Shouldn't be like, I mean, isn't like, if you have many genomes, don't you think that will be improving the overall scale of the graph? If you remove the low allele like mutations at the building of the graph.
Okay. So for sure, you could. It depends on the scale at which you're doing things. You're right that low frequency stuff adds relatively little value, and you can imagine removing it to make things easier. It's actually quite complicated for bacteria to decide what frequency means, because they've got strong population structure, and but this problem is like super easy with humans, okay? Because we almost know exactly where every human is on the planet. Like we can see them, we know them, we can sample from them. Their ancestral history is relatively simple. For bacteria, the unknown unknowns are enormous. Their strong population structure. RefSeq is unbelievably biased. So it's very hard to select a sort of unbiased doesn't mean anything actually, set of genomes from which to sequence to build the initial thing from. So there's what you're saying is right, that it's definitely valuable to try and balance the input data, but that is almost easier to to do at the point of sampling and when you choose like where your genomes are from, because a lot of it is about you know, the environment in which they're collected and things like that. But good point anyway.
Thank you.
Okay, I think I'm going to carry on, and we can come back to more questions later. If that's okay. All right. There's not much to say about genotyping. You sort of you genotype every site or subsite. Remember, you can have a SNP here and you can have stuff up here. I'm not going to talk much about that. It's just a simple personal model. Okay, and then this is the overall picture of what we're doing. So just remind ourselves, for bacteria, one reference is just impossible for reasons both of hard and soft bias. Pandora is our tool, which basically collects a set of local graphs or multiple sequence alignments. It addresses soft bias by using, you replacing a linear reference with a graph, and it deals with hard bias by having a set of graphs. And for each sample, what you're going to do is find the closest mosaic, use local assembly to find missing SNPs or indels, you augment the graph, and at the end you have sort of a complete representation of everything you have in all these samples, or as complete as you can given this graph, given the genes you started with, and then you go from there. So there is another issue, which is what you might call the representation problem. So the choice of reference genome when you're describing something in, say, VCF format, it really affects how it looks. So for example, I've got this SNP here between these two genomes, and I've got a black reference genome. And if I do represent this SNP in VCF format, then I have to give it coordinates on the black. So I have to start here. So I have to give this flanking sequence, then the SNP, and then this flanking sequence. So it looks like that. That's a pain. If I had chosen the blue line as my reference genome, then suddenly it really looks like a SNP. So this is sort of obvious. Normally, sort of in microbial genomics, you deal with this by you choose a reference genome in the same ST or the same clonal complex, or maybe if it's an outbreak, you can choose something from the same outbreak. But roughly speaking, you choose something that is pretty similar as much as you can. And what's going to happen is that how close it is will vary across the genome. So it'll work better in some places than others. Whoops. So what we want to do is we want to construct a data-dependent reference, which is closest to the set of genomes that we're going to compare. So Pandora, if you remember, inferred a mosaic approximation for each sample. It's the closest path for each sample. So imagine wiping the graph clean, and you draw on all of these mosaics for all of our samples that we want to compare, and you increment counts on the shared regions. And then use dynamic programming again to choose the most supported path. And now what you've done is you've chosen a reference genome that is as close as possible to the samples you actually want to look at. I will actually evaluate how well this works later on. So the goal is that the multi-sample VCF has the best reference for describing those samples. You want more SNPs and less long alleles. And of course, it's easy to annotate. It's just a gene. So there's no need for most users to engage with the graph. And that's just a general problem that, you know, graph genomes are fine and they're very elegant and all the rest of it. But there's a question of how you want to make this usable for people who actually want to take your output and study microbial genomes. So there's lots of strong reasons to support VCF in terms of community engagement, and that will make sense. As far as I know, this is the first time we've managed to support VCF without insisting the user provides a normal reference genome at the start. If you specify your reference genome up front, something from RefSeq, as soon as you do that, you inherit the hard reference bias of that single reference genome. So instead of doing that, what we want to do is produce a reference that depends on your data. Okay, so let's evaluate the results. These guys you've met. Leah is a microbial genomicist who's joined our group recently. She's done a lot of work on this. What we've got is we've got a pan-genome of E. coli. We took 350 RefSeq genomes, so it's about 20,000 genes, about 15,000 intergenic regions, which annoyingly we've taken from slightly different genomes, but maybe that shouldn't matter too much. And precisely because of the U-shaped frequency curve, you know, 60% of the genes and 40% of the intergenic regions are actually linear. There's only one allele okay, because there's lots of rare stuff. And given 20 gigs of RAM and 70 minutes, then we index the lot. And just a reminder, what are we trying to do? We're trying to find all the SNPs between these genomes. So we take 20 E. coli from across their phylogeny. Each of them has got matched Illumina and nanopore, and we've got polished long-read assemblies. We mask them to hide positions where the short reads don't support the assembly, and what's left is a set of the best truth we can get. And we're going to use them to find true SNPs and indels between the genomes. How do we do it? Well, hold on, let me show you our samples first. So this is an E. coli phylogeny. E. coli folk talk in terms of phylotypes. So each of these clades, I've just drawn the same thing in a different way. Each of these clades, A, B1, B2, D, and F, are sort of known E. coli phylotypes, which are things that E. coli people know and love. From our point of view, we just made sure that so each of these dots in the left-hand column are the samples that we're studying, and we've got them spread across the whole of the phylogeny. And then we also selected a bunch of reference genomes from RefSeq to use for the mapping-based callers we're going to benchmark with. Some of them are selected manually by just finding a RefSeq sample that's as close as possible on the tree. And some of them are selected by finding the closest MASH genome to each of our samples. So for each of our 20 samples, we found the closest MASH genome and the closest phylogenetic genome in RefSeq. Oh, oops, I keep doing that. So what are we going to do? We're going to find all the true SNPs between those 20 genomes. That's a huge pain. So we've done all pairwise alignments between the 20 genomes, generating truth SNPs. There's a bunch of work making sure that those are all really true. And then we have to decide if a SNP between genomes 1 and 2 is the same as a SNP between genomes 3 and 4, and the coordinates are all, all over the place. So we make a different graph. This is purely just for the evaluation. Essentially, a node is a SNP between genomes I and J. So this one here, for example, within some well-aligned region. And I join two nodes if they share a genome and a position. So for example, this is one node and this is another node, and they're connected because they share a genome and a position. And you find connected components within these, and these are equivalence classes of pairwise SNPs. So that's a very long, it's a pain. You shouldn't need to think about it really. All we've done is we've deduplicated all these SNPs to make sure that this AT refers to the same AT as that AT. And in total, we find about nearly 700,000 differences between our 20 genomes. And we want to check whether or not we can find them. There's one other thing. We have to check, like, how do we decide if a call is correct? So you've got a bunch of reads here and a genome and a SNP there. What we do is we take the variant and we take the flanking sequence. We take that probe and we map it to the truth assembly. And so it maps here. This is the variant. And if that maps correctly, I'm not bothered about a mismatch in the flank, so long as the flank is long enough that it's a very confident mapping. So we've actually got 100 bases on either side. And here, if it maps perfectly, but there's a disagreement specifically in the allele, then it's a false positive. Okay. How do we quantify this? So supposing here's a variant ACAGT going to TCTGA. That's actually 3 SNPs there. So if Pandora called it as TCTGC, so in other words, it was wrong here, there's one SNP that's wrong. And you could measure this 2 ways. You could say my error rate's a third because 1 SNP out of 3 is wrong. Or you could say my error rate's a fifth because 1 base out of 5. So because we're going to evaluate every position, and including whether or not we've made reference calls correctly, we're actually going for option 2. You could do either, but so long as we're applying the same error metric to all of our tools, I think that's okay. And then last of the preamble things, what's recall? Supposing I've got a gene which is present in 2 of my 20 genomes, so I've got an AT SNP between those 2 genomes, and the caller let's say the caller spots both alleles, so it's found a SNP. And then I've got another gene which is present in half of my genomes. Each allele is present in 5 genomes, so it's at 50% frequency. And my caller spots 1 G and one C and 1 G, and it misses 8 alleles. And now the third gene is present in all the genomes. It's a core SNP. Sorry, it's a core gene. And let's say there's 15 A's and 5 G's. And the caller spots all the G's, but none of the A's. So there are different ways you can summarize. It depends what you care about, right? So pan-variant recall, what I'm calling pan-variant recall is how many of these unique SNPs have you spotted? you need to have seen both alleles at least once, but you don't penalize for missing alleles. So here, I get a 1 for this guy because I have seen the A and the T. I get a 1 for this guy because I have seen the C and the G, even though I missed the 8. And here I get nothing because although I've spotted 5 Gs, I haven't spotted an A. So for this measure, I get 1 + 1 + 0 over 3, which is 2/3. That's fine, but I'm not very comfortable with missing all those alleles in the middle, and I don't really like that. So instead, what we're doing is we take this other measure, which is called average allelic recall. I mean, we use both. What percentage of the alleles for each SNP are spotted, and take an average of that. So 2 out of 2, because I found both alleles for the first one, 2 out of 10, because I only found 2 out of 10 for the second one, 5 out of 20 for the third one, and you average those. Okay, so here, now we sort of start getting into sort of the interesting stuff where we look at the results. This is a plot of average allelic recall on the y-axis and error rate on the x. This is Pandora's results. We've just taken 4 of our samples to evaluate because it's a pain re-base calling nanopore data. So we've base called the 4 both with normal guppy and with methylation aware. And here in blue are the results with just Pandora when you find the nearest mosaic. And here when you add de novo assembly of the reads, local assembly. And so you move to the right. So in other words, your error rate goes up from 0.5% to 0.65%. And your recall does go up a bit, but it's not massive. If instead you use methylation aware reads, you start here and you end up here. So your error rate has gone down and your recall has gone up. It's a win in every sense. So from here on in, we're only going to use methylation aware base calling for this data. And for those who don't know about nanopore data, how do I summarize this quickly? So you basically push DNA through a pore. You detect changes in electrical current based on like which base is going through. Those currents are actually also affected by the methylation state of the sequences it goes through. And they use neural networks to basically decide which bases are going through. Now, those networks are much better trained for some species, so for example, E. coli, than others. So it makes a big difference for E. coli. And I've just said that. Okay, so now if I look at all 20 of my samples, again with nanopore data, this is Pandora over here on the left. I can get just over 85% average allelic recall, and the error rate is about 0.5%. You can't really see on this scale because I had to fit all of these other guys on. So here we've got Nanopolish, standard tool, but using the 20 reference genomes that we selected from the phylogeny, and 24, sorry. And here, Medaka. All of these results are unfiltered. What happens when you filter is things move a bit to the left, but the general picture stays the same. But I wanted to see, one of the key things I want to see is how high you can get in terms of recall. Okay, and then that's recall. What happens to precision? And precision's there on the x-axis in some sense. But what I want to know is, I want to understand how precision varies with choice of reference genome for each of my samples. So I plot this a bit differently. Along the x-axis, I've got each of my samples, so each of my 20 samples. Y-axis, I've got precision. And then I've got box plots for each of my tools. So here, this is Medaka down here. Depending on, this box here is the set of results I get from Medaka on this sample using the 24 different reference genomes. So there's a huge variation here between sort of all, down to almost 50% precision up to sort of just over 80. So that's Medaka again, unfiltered. So you can move these up. And then the same here for Nanopolish. And up here at the top, Pandora only, doesn't depend on a reference. So there's only one line there. So that's interesting. How different is it for Illumina? Okay, so here I'm starting in the middle with Pandora. This is Pandora, just the mosaic, and this is Pandora with the mosaic and local assembly. So you can increase the recall and you don't affect the error rate again. Over here, we've got SAM tools, again, unfiltered, using different reference genomes. And over here, I've got SNPy, which is a standard tool everyone uses, which is based on freebase. And it does much better than we do in terms of error rate, much better. But has broadly the same sort of ceiling on recall based on reference genomes. if I do, I'm not sure if I have the plots on this slide deck, but if I did add some sensible filtering, then what happens is this guy moves over to the left, this guy moves over to the left, this guy almost doesn't move because it's almost as low as it can go. And it's a bit unfair to compare SAMtools and SNPy, because SNPy intrinsically does some filtering that, you know, it does some defaults of filtering, whereas SAMtools haven't done that. And there's no filtering on Pandora at the moment. And again, does the reference affect the precision? Yes, it does. So these are the results for SAMtools across the different genomes. This is Pandora here, and this is SNPy beating Pandora on precision up here, but also affected by the genome, the reference genome. So why does SNPy do so well? Well, SNPy is basically a reference-based graph or haplotype caller. It's designed to fix soft bias, so it does really well on precision. That's exactly what so SNPy or Cortex, which does assembly. I mean, all those methods which fix soft bias do really well on precision. Okay, so that's all good. I've basically introduced the method. I've given you some like broad statistics. I want to remind you what we're trying to do. We're trying to find all these SNPs. We're trying to show that we can find SNPs in rare or accessory genes. So at the moment, I've not shown you whether or not that works. So let's look. This is recall across the locus, a gene or intergenic region frequency spectrum. So this is the number of samples a SNP is seen in. These are box plots for SAMtools and SNPy, depending on what reference genome you use. And the dotted line is Pandora. So let's work our way from left to right. At the rare end of the spectrum, we're calling really tens of thousands of SNPs that are invisible to reference-based approaches. In the middle, we're sort of doing the same. And then over on the right, over here, you're getting, we're not doing as well in the core, essentially, as the standard reference genomes are. Now, this is absolute count. Remember, I said at the start that the frequency distribution of genes sort of looks like this. But what I'm doing here is I'm counting SNPs. So there's an open question, like how many SNPs are there in all of these places? Like how well are we doing in terms of as proportion of all the SNPs that could be discovered? So if I normalize, then it looks like this. So Pandora is calling 50% of all the SNPs that are present in just 2 genomes. And the reference genomes are calling sort of 30%. So this is our win. This is why we built Pandora effectively. I mean, we'd like it to be up here, and I can talk about that. But this is why it was worth it essentially. And over here, we have a gap in performance. We're not doing as well on the core genome, which I don't like, and I can talk about in a minute. But I'm not so worried about it, essentially because we've got plenty of methods for doing core genome calling. Now, oh, what does that mean? Oh, that's very annoying. My figure's gone. Can you see a figure? I wonder, okay, I'm sharing my screen, so I think I should be able to, hold on a second. Here we go. This is a bit silly, but I'm going to show you the figure here. This is a heat map of, can you see this heat map? Someone,
yes.
Great, thank you. Okay, this is a heat map. These are my 20 samples. And what I'm doing is I'm counting how many 2 SNPs, the SNPs that are only present in 2 genomes, how many of them are shared between each of those samples. So I want to know if the SNPs that are only there in rare loci, what I want to know is if they're enriched inside phylotypes, okay? And what we're seeing is, and this is a log scale of counts of SNPs, and what you can see is that they're just everywhere, okay? You're not seeing an enrichment only in purple and purple. You're not seeing, they're just everywhere. And so basically these rare SNPs, which are, we expect them to be basically there in mobile elements and essentially mobile things that sort of keep moving into and out of the population. Those are not so constrained by the phylogeny. And from my point of view, this is a great advantage for what we're doing, because I want to be able to compare diverse data sets that I don't want to be limited by only looking at things which are very similar. You can look at this another way. So references work better for genomes which are in the same phylotype. So each of these histograms shows SNP results with a fixed reference. I've got the evaluation of 5 different reference genomes, and these are SNPs results for the 20 samples. And the background color is the phylotype, the clade, in which the reference genome sits. So here I've got a purple reference genome. Here's the purple clade over here. I've got a purple reference genome, and it does really well on recall for the purple genome. So in other words, a phylotype A reference genome does well for phylotype A. So let's skip yellow for a second. A phylotype blue, a B2 reference does well for B2, and same for green and red and red. Couple of caveats. Yellow is an outgroup. You don't have any samples inside yellow. Nevertheless, it does reasonably well for pink, which is quite close to it. And then over here, red and green are sort of in the middle. And although, as well as doing reasonably well for themselves, they do quite well for each other. So that's interesting. So, and that's, you know, no microbial genomics person is going to be very shocked that if you choose a reference from your own phylotype, you'll do a bit better. What's interesting is if we now restrict to the rare genes again, so again, I'm going to look at only at SNPs that are present in 2 genomes. Whoops. Ah, come on. There we go. Now I've got the equivalent thing underneath, but now I'm only looking at the SNPs that are there only in 2 genomes. And there, I'm sorry, I should have said that the black line is Pandora's performance, which is sort of flat. And here, it's a bit more spiky, but it's still, we do consistently better than everybody else, which is by design, of course. So I'm getting towards the end. Just remind ourselves what reference is Pandora using inside its VCF. We took the mosaic approximations of all our 20 samples, we overlaid them, we did dynamic programming to find a VCF reference as close as possible. So how well does that work? Is it really closer to the samples? So what do we do? For each gene in each of the 20 samples, compare it with the 24 RefSeq references and compare it with Pandora's VCF reference. And we measure the edit distance scaled by gene length. And we ask, how often does the references version of a gene sit within 1% of the samples version? I want to know how close the references are locally compared with Pandora's version. And this is what it looks like. Here's the frequency along the x-axis, and this is a log scale, and it's a count of genes which have got edit distance less than 1%. So, and here is Pandora up here, like doing orders of magnitude better than any of the reference genomes over here. And basically it does better all the way up to 15 samples. So it's doing a good job of finding closer things. Okay, and then, so you may need a break. I've covered essentially all of the main content. There's one thing that's worth highlighting. If I compare Pandora on Nanopore and Illumina, Nanopore's got much worse error rate than Illumina, and this is weird, okay? The first time you see this, this is weird. Look at the error rates, they're basically the same. So if you look at the top right of this curve, so this curve is, again, it's error rate here on the x-axis, it's recall on the y-axis, and how you move down this curve is you sort of, we have a concept called a genotype confidence. Basically tells you how confident you are about a SNP call. And if you demand that you're more confident, you get a lower error rate and you get a lower recall. So that's not very surprising. What is surprising is that with Nanopore data, you have slightly higher recall for a start. You're over 75, well over 75%, whereas this is just over 74. And the error rates are, you know, pretty close. This is just over 0.6%. This is 0.58%. And that is, on the face of it, kind of unbelievable. Like, how is that possible? You know, what's going on? So I think the thing to remember is the graph is a strong triad. It allows us to zoom in on a close approximation, and it's not affected by the Nanopore errors. You can't, even if there are errors in the Nanopore reads, you're forced to choose a mosaic that's already in the graph. And there is no Nanopore sequence in the graph. It's built from pre-existing genomes. So that's the first component. So you expect our initial mosaic to be good. Second thing is mapping non-Nanopore reads is easier than Illumina reads, and it allows better filtering of bad minimizers because you can with a nanopore read, which covers multiple genes, you can be sure where it comes from. And having decided that it's from a particular gene, you can throw away particular minimizers as being errors. On the other hand, with an Illumina read, which is only 100 bases or whatever, you could be less clear about where it comes from, and you're less able to throw away sort of errors. So there's that, and there's of course the fact that nanopore only does this well if you use methylation web-based calling. Okay, I'm just going to remind us what we've just done, and then I'm going to stop. And I'm going to remind you also, this is all Rachel's and Michael's and Leandro's and Leah's work. So what do? We represent everything as a DAG of minimizers. We use this collapse, a recursive sort of collapse and cluster algorithm to build our graphs. Our Pan-genome is a union of small graphs. It's driven by biology. Like we break up into genes and intergenic regions, and it allows massive parallelization. The construction from the multiple sequence alignment, it preserves more haplotype information in complex regions. So it avoids the blow up problems that many graph genome methods struggle with, and it supports linear nanopore. Single sample inference, you just quasi-map your reads to the graph, try and work out your best path, do local assembly to do a bit better. And then multi-sample comparison, you do dynamic programming on the collection of sample paths to choose your VCF reference, one path per gene or intergenic, whatever. And then indexing, so there is a downside to this whole approach, which is indexing. You know, if I'm doing BWA mapping of a genome, I index the reference once and that's it. And it could be slow. I mean, it's not slow with BWA, but it could be slow because you only do it once. In our case, we need to augment the graph with all these novel things we found from local assembly in the middle of our workflow and re-index. And that's the slowest step in our process at the moment. And again, a reminder, if you were to choose just a RefSeq genome as your VCF reference in advance, you immediately accept hard reference bias, which is what we're trying not to do. So having said all that, you can't escape biology. Okay, that universal gene frequency curve tells us no linear reference is enough, but also no pan-genome reference is going to be enough either, because it's built from a fixed panel and there's always going to be rare stuff that's missing from your panel. Now, from my point of view, that's fine. I sort of think of this as a bit like, it's a bit like an English dictionary. There's always a trade-off between utility and comprehensiveness. I could have a massive dictionary that I can't lift, which has got everything, or I can have a lightweight thing that mostly functions in most cases. If you really want to, and you don't want to lose anything from your cohort, run PROC or whatever you like, get the genes into intergenic regions, build a dataset reference graph, and then you're able to access absolutely everything in your data. But you're able to do it in this way that allows you to access and describe and compare all of your genomes. So what are the limitations? RCC construction algorithm, the collapse and cluster thing with that animation, that's at the moment in Python. And we're using cluster Lomago, which is really not the fastest multiple sequence alignment algorithm. So that augment stuff is slow. It's 8 hours at the moment, dominated by about 12 genes that are very slow. That needs re-implementing in C++, which is what's just about to start. We build a PRG from RefSeq, and that would be much better to use a well-sampled data set. And really what you want is samples that are collected by, by ecology, by environment, by host, if that's appropriate, as well as bearing in mind the phylogeny. So what you want to collect as representative a data set as you can. And then finally, I mean, the thing that really bugs me is that there's no reason why we should have a worse Illumina error rate than SNP. I need to explore that. I sort of half suspect that there's some underlying either curation bug in the way that we got our input MSAs or something like that. I need to check into that. So what does it open up for us now that we can do this? Well, microbial genome-wide association studies are now going to be able to do, associate actual SNPs in accessory genes instead of either doing presence, absence, or KMAs. So it affects all of genopheno analyses, I think. Population genetic models of bacteria, I mean, that's very hard, but at least this allows you can basically convert all of this into zeros and ones, basically, which is the sort of input data from many population genetics methods. And it allows you just to study diverse data sets. And so our conclusions, well, variant calling bacterial genomes is not the same as in eukaryotes. I think you knew that. The frequency distribution of genes means there's a lot of variation that's currently inaccessible. Pandora recovers a lot more. How much more depends on how you construct your graph and how it compares with your data set. It's still a work in progress. The augment and compare steps can for sure be dramatically accelerated. We focus very much on being correct rather than fast for the moment. And the preprint is basically ready, but we need to make some sample. We need collaborator permission to make our samples public. So you should see that in the next couple of weeks, I hope. And that's all for now. Questions, anybody?
All right, thank you, Dr. Iqbal. If we're going to go off the previous format. So if anyone has a question, please use the raise hand feature and I will call you out to speak. Yes, Carolyn.
Hi, hi Zam, great, totally fascinating. Okay, so as you know, we have looked at trees. So take our multiple sequence alignment and then do inference on it and reconstruct phylogenetic trees and then try to tell stories about the multiple sequence alignment and how it evolved by reading those trees and coloring them in and looking at their shapes. Could we do that same process? So after running your first part and saying, now we're going to represent our multiple sequence alignment as these cool graphs where certain regions kind of expand and you have different alleles and paths through that. So have you thought about, interpreting those, or what are the kinds of patterns that you see in those graphs that represent your multiple sequence alignments? And how you might look at, could you look at within host or within sample diversity and kind of use those tools almost in lieu of phylogenetic trees to understand what your data set means?
Okay, there's a lot to unpack in there. No, it's great. I'll work backwards. So yes, definitely within host diversity was really where this was intended. Like we were dramatically unrealistic about how much we thought we could do in one PhD. But within host diversity was exactly where we wanted to go. And so the reason for these DAGs are exactly designed to be able to sort of pull out multiple paths effectively as well. So yes, we want to go there. I had not considered literally using the graphical patterns instead of trees. I have been, I have thought about how this would affect sort of people who do sort of ancestral recombination graphs and things like that. If you're thinking of basically using the structure of the graph itself as a replacement for a tree and then looking for patterns that way, that would be super interesting. I think if you want to do that, then that goes back to, I'm sorry, I've forgotten your name, was it Feroz? I mean, I think you also need to notice, at the moment we've collapsed if multiple samples take the same path. You probably want to notice that. Yeah, that would be cool to look at.
Thanks.
Okay.
Any additional questions?
I've not been looking in the chat. Ben has said perhaps Julia would be better than maybe. I mean, the whole thing is in C++. It's just that's the last component that hasn't moved into C++ yet. So I think we'd probably try and get it all into the same language.
I guess I perhaps have a naive question myself. So to what extent is the information in single variants useful in this type of work? And like, can there be gains made if you say use methods making use of like multiple variants in mind when you're calling variants in like a microbiome?
So okay, I guess my answer to your so your first question is like, how useful are SNPs anyway, essentially? And I think, I mean, not meant in a mean way, I know. I think my position is I want to be able to see everything. And there are definitely times, you know, particular mutations in antibiotic resistance genes affect the antibiogram. Changes in CRISPR regions affect, you know, what they react to. I've got, you know, embarrassingly, I've got more examples in the paper and I've forgotten them for the moment. But I mean, there are clearly going to be times when we care about SNPs. There are times when you just try and put it another way, people try and do just gene presence absence as a way of looking for correlations with phenotypes, and it doesn't always work completely. And I think some of that is because it depends on the precise sequence. So there's that. The idea with doing our funny block-like thing, but supporting nanopore, is that because nanopore covers multiple, a nanopore read is long enough to cover multiple of these genes, you can get gene ordering out of it. And although I haven't talked about it, we actually have a secondary graph where the nodes are genes, and you've got the graph is sort of, you know, which genes come after which in the reads. And I think, you know, where we want to be going is we want to be analyzing both at the level of structural changes, because inversions and, you know, recombinants and things like that, and at the SNP level at the same time. And yeah, you've taken things one order of magnitude more difficult than Caroline has, because you want to go. She was talking about mixed infections. You're talking about metagenomes, and yes, I mean that would be super interesting. And there's there's sort of no reason why we shouldn't.
Thank you.
Okay.
Yep. So, yes, we have another question from Carolyn.
It's me again. I think that last question was really great and really interesting in terms of, okay, so now I've run Sam's fantastic pipeline and I've got my SNPs, and for any two of my bacterial genomes, I can say how many SNPs apart they are, but I can also say in which genes, how rare those genes are, what are the, you know, some of the ecological roles of those genes, are those in drug resistance conferring genes, how have those genes spread around? So actually it seems like there's a launch of a fairly big set of questions around how you now interpret these data, from the simplest thing we might want to do, which is, hey, new isolate, are you in or out of my cluster? And if so, why or why not? All the way to looking at dispersion of resistance conferring genes and all of that. So I wondered if you'd thought about some of those interpretive questions.
I have. I mean, the honest answer is I've been a bit buried in trying to make it work, which is or at least, in fact, not so much to make it work as to get the evaluation working properly. And now that the paper is essentially finished, and I'm emerging again from the ocean, and it's exactly the kind of question I think we want to approach. I mean, really, all we've done so far is make it possible, and now there's a whole world of things we need to try.
All right. And if there is no one else, I'd just like to take this time to thank both of our speakers today, Dr. Iqbal and also Ben Cardone, and to give a thanks to our sponsors for VanBug. We're sponsored by the Bioinformatics training program at UBC and SFU. We're additionally sponsored by Genome BC, Ecoscope, and bioinformatics.ca, and also Abcelera and Langara College. So we're also affiliated with several other bugs throughout Canada. So go ahead and check those out if you're interested in attending more of these types of talks. Again, thank you, Dr. Iqbal, and thank you, Ben. And yes.
Thanks a lot, everyone. Take care.